So in lesson 8, we're going to start looking at the HL material and we're going to learn about a drug called Taxol and what that is used for. Here are the understandings as well as the applications. So a quick review on chirality. So as a reminder, a carbon atom is considered a chiral center when it bonds to 4 different groups. There are 2 different versions of the chiral center. They are either right-handed, which is given by the letter R, or the left-handed version, which is given by the letter S. These versions have identical physical and chemical properties, but in a chiral environment, they will react differently. One of the biggest examples is in the body, where enzymes only exist as one enantiomer. So let's review chiral molecules and consider why may only one enantiomer react in the body. So if you remember, chiral, 4 different groups attached, and you're going to have a mirror in the middle, and you have a mirror image. So if we look at the right hand image, you will see why it is so important um, that these molecules be considered as both enantiomers. So on the left-hand side, if we have the active enantiomer, okay, it is arranged in a certain way, and you will see that it'll react with the drug binding site and it'll fit perfectly. So A to A, B to B, C to C. But if we look at its mirror image, its inactive enantiomer, you can see how this molecule here is an enantiomer of the one on the left. And now they do not line up, right? B to B lines up, but not C and A. So therefore, this is an inactive enantiomer. It will not react with the drug binding site. So even if you rotate it in a certain way, it still won't line up perfectly. So it's very important that we get the active enantiomer for the drugs that we're trying to create. So biological synthesis reactions within the cells, okay, that's given by the term in vivo, produce only one enantiomeric form. So when a drug is harvested from a natural source, such as morphine from opium seed, only a single enantiomer is obtained. Okay, so that's in the body, within the cells, in vivo, one enantiomer. But when drugs are produced by synthetic processes outside the body, given by the term in vitro, they yield a mixture of enantiomers, which you may remember as a racemate. So, pharmaceutical companies then face the challenge of finding out the physiological effects of each isomer before determining whether the drug can be marketed as the racemic mixture or whether a single enantiomer must be made. As we'll see, some drugs can be marketed and used and given out as a racemic mixture, but sometimes only one enantiomer can be used. The other one could have some really bad negative side effects. One of the biggest examples of this is the drug thalidomide. The drug was initially manufactured and sold as a racemic mixture. We can see the 2 examples of the thalidomide with the chiral carbon center labeled on each one, so we see our mirror image. But it was discovered that only the R isomer, which is given on the left here, had the desired effect, which was to induce sleep in pregnant women. Its enantiomer, the S form on the right, was known as teratogenic, meaning that it caused serious deformities in the fetus. Okay, so only one enantiomer produced the effect that it was looking for. The other one was um, not good. So it is known that the two forms of the thalidomide drug interconvert under physiological conditions, so administering a pure isomer would not protect against the teratogenic effects. Drugs that are marketed as racemates include fluoxetine, which is known as Prozac, and the anti-inflammatory drug ibuprofen, which, like thalidomide, undergoes enantiomer interconversion in vivo, right, within the body. Okay, because of that, thalidomide, it is no longer used for pregnant women. So now let's talk about the main point of this lesson, which is Taxol, which is used as a chemotherapy agent. So chemotherapy means the treatment or control of a disease by chemical agents, and it is generally used in the context of cancer treatment. Taxol is sometimes known as paclitaxel and is one of a group of related compounds known as taxoids. It is used primarily in the treatment of breast and ovarian cancers. So let's look at Taxol and what it actually does. So Taxol's anti-cancer properties are a result of its ability to bind to a protein called tubulin in cells. So tubulin is the main component of microtubules, which form structures called spindles during cell division. When Taxol binds at the microtubules, it prevents the spindle fibers from breaking down, and this halts the cell division cycle. In this way, Taxol prevents growth of the tumor. So if you know anything about cancer and cancerous cells, if you have a tumor, then those cancerous cells divide and the tumor grows. You have increased cancer and the possibility of the cancer spreading. Well, if Taxol and other similar drugs bind to these microtubules, or specifically to the tubulin within the microtubules, then you could prevent the cells from dividing and that tumor growing and the spread of the cancer. Okay, you can see an image on the right of the drug Taxol, the molecular structure, how it attaches to the tubulin in the microtubules. So one of the challenges of designing this drug is that Taxol is a very chiral molecule. It has 11 chiral carbon centers. This means that it can exist as a large number of enantiomers, which may have different effects in the body, including the possibility of harmful physiological effects. Only one enantiomer has the desired therapeutic activity. That is a big problem. Making all the enantiomers and isolating only one is far too wasteful. Right? With 11 different chiral carbon centers, you have all these different possibilities when they go through the drug synthesis route. So there has to be a way to design this drug and create it so that we get the enantiomer that we want and need, but don't waste. Okay, so here is a picture of Taxol. It is quite a large drug with many different functional groups, and within this there are 11 chiral carbon centers. So how do we go about designing, creating this Taxol? So this is called enantioselective synthesis. It is therefore of more interest to pharmaceutical companies to find ways of directly synthesizing a single enantiomer. One strategy one strategy to achieve this uses a chiral auxiliary. So this is a chiral molecule which binds to the reactant, physically blocking one reaction site through steric hindrance, so ensuring that the next step in the reaction can only take place from one side. This effectively forces the reaction to proceed with a specified stereochemistry. Once the specific enantiomer of the new product has been set, the auxiliary can be taken off and recycled. So, during the synthesis of molecules using all of those organic reactions, sometimes when you perform a chemical reaction, you have two possible outcomes based on the chirality of the carbon centers. Well, if you can block one of the two reactions or outcomes, then you will only get the one that is desired in a specific step. So we do this through the use of a chiral auxiliary. It is put on at one step during the reaction, then it can be removed later on and reused. So here is a simple example. So let's say you take propanoic acid, and if we look at the top route, let's say we don't use a chiral auxiliary. If we do a reaction which adds an amino or an amine group, okay, at our chiral center, you have two possibilities, okay? And in fact, both enantiomers will be produced, okay? This one here is coming towards you, the NH2. This one here, the NH2 is going backwards, okay? Well, let's say we don't want both enantiomers, we only want one of them, okay? So if we add a chiral auxiliary, let's say we place some sort of molecule at the end, and therefore we will only get one enantiomer. We will only get the NH2, the amine, that is coming towards you. Okay, by placing this chiral auxiliary, you force the reaction to occur in the manner you want. And then at the end, this auxiliary can be removed, and it can be replaced with this OH group. And therefore, we only get one enantiomer, which is the one that we want, the one that we need. So let's look at some of the issues with Taxol other than the fact that it is a very chiral molecule. So because it is so complex, its commercial synthesis from simple molecules involves about 30 steps and has such a poor yield it is impractical. So with every step you get waste because you can only use certain products, and therefore the amount of Taxol you get at the end just isn't worth the price and the process. So a more efficient process now uses the extraction of a compound which is related to Taxol from the needles and leaves of yew trees from Europe and the Himalayas. Harvesting the leaves does not damage the trees in a way that removal of bark does, and so this is a more sustainable approach. The related compound, known as 10-DAB, is then chemically modified to form Taxol. The process is known as semi-synthetic synthesis, as it starts from a precursor obtained from nature. Okay, so taking the leaves from this yew tree, okay, it has a compound that is very similar to Taxol, and with a few modifications, you get the drug you want. So it's not purely synthetic, it is semi-synthetic. You start with something from nature, do a couple of synthesis reactions to get the product you want. And the very important thing is it does not damage the trees because you're using the leaves and not the bark. So going back to the idea of enantiomers, while the enantiomers of the optically active substance can be distinguished using a polarimeter because they rotate the plane of plane polarized light in opposite directions. So if you remember, the different enantiomers will rotate light the same amount of degrees, but it'll be in opposite directions. So the R form will rotate it one way, the S form will rotate it the opposite. The simple polarimeter consists of a source of light, two polarizing filters, a sample tube, and a scale to measure the degree of rotation of the plane polarized light. If the enantiomer rotates plane polarized light clockwise to the right, it is called the positive enantiomer, and the one that rotates the plane anti-clockwise or to the left, it is called the negative enantiomer. This has to be determined experimentally. You cannot just look at an enantiomer and determine which way it will rotate the light. So let's summarize what you need to know regarding Taxol. Okay, so you should know the natural source of Taxol. It occurs naturally, mainly in the Pacific yew tree, but only in very small amounts. Even if this tree is grown commercially, it cannot produce all the Taxol required for therapeutic use. The main thing with the structure of Taxol you should know that it contains 11 chiral centers. How does Taxol work in the body? What are its chemotherapeutic properties? Well, it is effective against several different types of cancer. Most specifically, we mentioned before, the ovarian and the breast cancer, but also lung and pancreatic cancers. And it works by interfering with tumor cell division by binding to different proteins called tubulins. We talked about the semi-synthesis of Taxol. So it can be semi-synthesized from that 10-DAB, which is a precursor of Taxol. Remember, it has a very similar structure. One of the big things you have to know about the process of creating Taxol is the use of chiral auxiliaries. Okay, remember that if you want to get one form, you add a chiral auxiliary to force the reaction to go in one direction. And then last but not least, we can test the drug for purity using a polarimeter by looking at the plane polarized light and see to what degree the light is rotated. All right, just a couple questions to wrap this up. Describe the original source of Taxol and the environmental impact of obtaining the drug from this source. Okay, so you can check back to where we can get our natural sources, and that would be the bark of the Pacific yew tree. Harvesting has depleted the trees which grow slowly. All right, let's look at letter B. Taxol has been described as a very chiral molecule. Explain the meaning of this statement and why processes to synthesize Taxol chemically are complex and must be carefully controlled. Okay, so think about, why is Taxol very chiral? And then why do we have to be very careful with this, and why are the processes complex and must be carefully controlled? Well, Taxol has 11 different chiral carbon atoms, giving rise to a very large number of possible stereoisomers. At many stages in its synthesis, different enantiomers could be produced which have different physiological properties, so these steps need to be controlled by chiral auxiliaries. Okay, if you have all these different products, they might not produce the effect that you need in the body, the physiological properties. All right, last but not least, Describe how chiral auxiliaries can be used to synthesize only the desired enantiomeric form of a drug from a non-chiral starting compound. Explain why it is important to use only the desired enantiomeric form of a drug, and give an example of what can happen if a racemic mixture is used. Okay, so hopefully you remember from before what a chiral auxiliary is, what it's used for. So it is in itself an enantiomer which bonds to the reacting molecule to create a stereochemical environment necessary to follow a certain pathway. So you have two options, but you force it to go in one direction. The reaction then takes place, forming the desired enantiomer, and the chiral auxiliary is then removed. So it also asks why is this whole process important, and what can happen if a racemic mixture is used. So different enantiomers can have different biological effects, some of which may be harmful. And one of the biggest example was the genetic deformities caused by the S enantiomer of the drug thalidomide in the racemic mixture.